Okay, everybody, you can find a seat. So thanks very much for coming back. I have the pleasure of introducing my boss, because Oliver House has taken over as the head of the Department of Psychosis at the Institute of Psychiatry, so I have to be very nice to him. But I would be nice anyway, because Oliver is currently the most cited and influential person at the Institute of Psychiatry. And really over the last 12, 15 years, he's produced a whole succession of wonderful work on the molecular pathology of particularly psychosis, but also, as we heard this morning, had a look also at serotonin and depression. So lots of studies on dopamine and glutamate, and more recently also on inflammation and synaptic pruning. So I guess from Oliver's point of view, the difficult task may be deciding which study to talk about. But you have quite a lot of so we have you before and then after the break. So yes, sir.
Thank you very much for a kind invitation, Robin. Remember, it's appraisal season, so no difficult questions. So it's a great pleasure to be here in the room of the Spanish Inquisition, slightly nervous. I'm going to talk in the first half about a bit about the pathophysiology of schizophrenia and psychosis. And as you heard from Robin, I've done a lot of work on dopamine. Actually, it's not there, but my middle initial is D, and some people say that my middle name is actually Dopamine, although I'm not going to reveal what my real middle name is. So I want to start with a few disclosures. As you know, as you've heard, I work at the Institute of Psychiatry in the Maudsley Hospital. I've just finished a stint as Vice President of well, that's actually translational. It's not trans as in transgender neuropsychiatry at Lumbeck, and I've worked with a number of companies. But I'm not talking particularly about treatments in the first half, but bear that in mind for the second half, of course. I don't have any shares or any investments in any of these, sadly. So the other thing I need to disclose is that I come from a country that votes people like this in as our Prime Minister. And why is that relevant? Well, of course, his government in the run-up to Brexit said they don't believe in experts, so maybe you shouldn't bother listening to me at all. I'm sure Boris would say that. But that's the other disclosure. Then something else I want to highlight right at the beginning is lots of people that have worked with me on the studies that I'm going to talk about today, or this particular session, really want to highlight Stefan Brugger, Samir Jauhar, Rob McCutcheon, Ellison Wordy, Michelle Coquinu, as having done a particular contribution. And of course the patients and volunteers who've taken part in our studies. So I want to ask you a few questions as we go along, because I know you've had lunch and that can put people to sleep, so I need to keep at least the upper part of your body moving. So I've got a very high-tech question and answer system, and it's something that we need to check now. So that involves putting your hands up, very high-tech as you can see. And I'm going to start it with a nice uncontroversial question to begin with, to see if your arms are working. So who should have won the Eurovision Song Contest this year? And your choices are Sweden, Italy. No, it's good that you're responding, but wait, I'll give you the choices. UK, Silvio Berlusconi, don't care. So who wants to vote for number one? Oh, some Swedes. And Robin? Italy. Oh, lots of Italians. I heard it was good. 3, the UK. Some UK? I have to say I didn't understand the UK's entry. Silvio Berlusconi? Oh, there's one person. Don't care. There's lots of people. Okay, we'll come back to Eurovision, actually. There's a little theme in my talk. So I'm glad the Q&A system is working. Now a proper question. So this is something that we've just been looking at. So how many drugs have been licensed for psychiatry by the FDA, which is the regulator in the USA, in the most recent decade that you can get information for? So this is a question for you all. I'm not expecting you really to know the answer, but what do you think is the what sort of ballpark? So in 10 years across the whole of psychiatry, how many new drugs have we had? First choice is 87, second is 12, third is 52, and the fourth is 28. So who thinks 87? First hand. Be brave. Yes? Okay. One person. Number 2. Who votes number 2? There's a few people. Number 3, 52. One person. And number 4, 28. There's a few more. So I would say actually number 12 probably shades it, followed by 28. Right. Do you want to see what the answer is? So this is something that we just published. Luke Baxter did this with me. We looked at the FDA database. So first of all, in oncology, how many do you think? Whole cancer in 10 years. Yeah, well over 100, nearly 140, 138 I think it was. What about neurology? Neurology doesn't do so well, but still a pretty respectable 52. Psychiatry, 12. So some of you already knew the answer, even though perhaps you've read my paper, but yes, it's very sad really, isn't it? 10 years, and we've only had basically one drug a year across the whole of psychiatry. I'm not just talking about psychosis, so this is depression, everything else. Of course, sometimes people say, well, perhaps the last decade was a bad decade. But actually, we looked back over the previous 2 decades as well, and it was the best decade. This is as good as it gets. So why am I telling you all this? Well, I'm telling you this because this is why I think we need to care about the pathophysiology, because we need to understand what's going on to try and develop more and better treatments. As I think we can all agree that the lack of treatments is not because there's not a need for things that work and work better. So that's my intro. So now we get on to a bit about dopamine. The dopamine, of course, has been around as a hypothesis of schizophrenia for many, many years. I think this is probably the oldest and proper account of a dopamine hypothesis, written by Stephen Stahl and Herb Meltzer. We'll come back to that. Robin's nodding his head, but I'll discuss that later. We'll come back to Stephen Stahl in a bit. Now, I think of this as a bit like, the dopamine hypothesis is a bit like ABBA. Because just like ABBA, it came on strong in the 70s and hasn't gone away. But I think the big difference is that the dopamine hypothesis has got some new material, whereas ABBA just recycles the same hits. And of course, this is relevant for the Eurovision contest because they won Eurovision in 1974, which has led them to their later success. So, question for you now. Where is the major dopamine abnormality in the brain of people with schizophrenia? And Robin's not allowed to answer because he's probably seen this before. But the choices are, is it increased D2 receptor levels, increased D2 receptor density, reduced dopamine transporter levels, an elevated dopamine synthesis capacity, or increased D3 receptor levels? So a moment or two to think about it, and please think about it online. I won't be able to see your arms going up or down, but you can still do it. Marta's going to check. So if you think 1 is the right answer, please put your hands up now. No one. What about 2, increased dopamine D2 receptor density? No one. Oh, the Q&A system, maybe it's broken. 3, reduced dopamine transporter levels. There's a hand, 2 hands, 3 hands, 4 hands. 5 hands. The Q&A system is working. 4, elevated dopamine synthesis capacity. Few hands going up there. And 5, increased D3 receptor levels. There's 1 or 2 hands. So I think I should have had another option, which is don't know or don't care. So how many wants to put their hand up for don't know or don't care? I won't ask you which. So there's a few of those as well. So that's interesting. So I'd say 4 and don't know or don't care are probably the ones that got the most votes. Well, I'm going to just walk you through quickly the data that we now have, because we now have lots and lots of studies, more than 60 studies that have looked at different aspects of the dopamine system in people with schizophrenia using imaging. This is showing you what PET imaging does. You don't actually explode something in someone's brain, but that's showing that there's a release of a gamma ray which allows you to localize receptors and proteins and enzymes. And with the dopamine system, we have lots of different approaches to measuring it. So we're very lucky. We can measure receptors, can measure the synthesis of dopamine, its conversion from tyrosine and dopa to dopamine. We can measure transporter levels, etc. So this has been done extensively. And in fact, people first started looking at the postsynaptic receptor, the D2 receptor levels. And actually, I said that the dopamine hypothesis went back to the 1970s. Well, you could argue that it goes back a lot longer than this, because this guy, Shakespeare, wrote about it. Did you know he was a schizophreniologist? Did anyone know that? No? Well, I'm going to try a joke in Italy, which only really works in English. But, of course, he famously wrote, D2 or not D2? That is the question. You have to laugh now because the jokes don't get any better. So what is the answer then? Was Shakespeare right that D2 is the answer to schizophrenia? Well, sadly, he was wrong. He got a lot of things right, but on this it's wrong. This is a meta-analysis of all the studies. You can see lots of studies done over the years. 0 means there's no difference between people with schizophrenia and healthy individuals. And you can see that with the exception of 1 or 2 studies, it's pretty much on 0. P-value is 0.05. So there might be a small increase. It's not significant. And Shakespeare had to change his famous saying to probably not D2 is the answer to the question. But as I hinted, there are perhaps 1 or 2 studies that do show higher levels. So is there a subgroup of people that have higher D2 levels? Well, I think the jury is out there, but this is potentially very clinically relevant, because if you do give antipsychotics to people, to rats, you see an upregulation in D2 receptors. Now, no one's actually tried to measure this in people taking antipsychotics, but there is a study that looked at levels of D2 receptors in people who had been taking antipsychotics for a long time and then stopped them and compared those to people with schizophrenia who had never taken an antipsychotic, who you see here in the see-through bars. So this is the level of D2 receptor availability in those people prior to any antipsychotic treatment. And you see their levels are around about 2.5, which is the sort of level you get in healthy people as well without schizophrenia. And then you see here this group who had taken antipsychotics for some time. And they'd not just taken antipsychotics, but they actually had either breakthrough psychosis or developed tardive dyskinesia and other side effects. So they're not representative of everyone who's taken an antipsychotic. I want to stress that. But what is interesting is that they had higher levels of D2 receptors than those who'd never taken an antipsychotic. That's the mean there. But something not commented on the paper, which I think is very interesting, is that even in this group, there's a big variation, and some people didn't show any real alteration, whereas 1 or 2 showed quite large elevations. This is double the average. So there's some variability in your D2 receptor system, and we just don't know if this is predating their treatment with antipsychotics or if it's linked to it. But I would guess it's linked to it because when you look at drug-naive patients, you don't see it. Now, I think this is really clinically important because I think it could underlie breakthrough psychosis that you get on people taking antipsychotics long-term. Unfortunately, it's not a very sexy area, so funding bodies are not interested in us doing the sort of study that would really test this. But bear it in mind when you think about patients who have started to getting breakthrough symptoms, that maybe you're chasing an effect of the drug. Because what we, of course, do is just put up the dose, but that doesn't tend to work. In the long term, they start to get breakthrough symptoms again on the higher dose, and then you put it up again, and then it just doesn't work. Maybe this is what's going on in some people. So, D2 receptors not altered by and large in drug-naive patients. But one other thing you need to know about D2 receptors is that they change over age, which is all a bit sad for people like me who are beginning to get on and long in the tooth. And this is a study that's just come out that quantifies this in healthy people. So this is the D2 receptor levels in 20-year-olds, and then you see in 70-year-olds. And you can see that there's a decline over time in the striatum in men and women. I mean, interestingly, it looks like it's steeper in men, but actually that's because you've only got a few older men, and it could be driven by that. So I think they need more data. I'm not accepting that men age more poorly, but what the take-home message, of course, is that there is a decline over time between around about 5% a decade. So by the time you're in your late 50s, early 60s, you've got quite substantially lower D2 receptor levels in your brain. Why does that matter? Well, it matters if either you develop your psychosis in your 50s or 60s, and your doctor is choosing the dose to put you on, or if you've been on treatment long term, because of course you may not need the same dose that you had when you were in your 20s. And what's the evidence for that? Well, a colleague of mine who used to be at the IOP, now in North London, she's very interested in this. This is Suzanne Reeves' work. She's looked at D2 receptor occupancy on amisulpride in older adults. So this is the relationship. So you see this nice relationship between D2 occupancy and the concentration of amisulpride in the blood. That's what you would expect. What dose of amisulpride do you think you need to get 60 or 70% D2 occupancy in someone in their 60s or 70s? Anyone? 50 milligrams? Anyone? Well, how much do we give to someone in their 20s of amisulpride? 400 twice a day? 400 twice a day. Well, 25 milligrams is enough for some. Marta's very pleased because she was in the right range, and you were. So in these people in their 70s on average, you do not need high doses. And that's probably because their D2 receptor levels have declined so much, and you just do not need that high sort of dosing. Marta?
Oliver, I know you don't believe that men decline faster, but say this is actually true. Could you explain women's second peak of onset of psychosis, or contribute to that?
It's a really interesting question, because women, of course, get that small second peak in the instance of psychosis perimenopausally, and estrogens do have effects on the dopamine system, so there is speculation that could be part of that. And yes, maybe women their risk remains a bit higher than men in the long term because they don't have quite the same drop. But as I said, look at the data. It is driven by one or two men, so it's declining in men and women. But I wouldn't read too much into that. But it's a good point. The basic message, though, is that we need to rethink what we're giving people over time, because what you had when you were in your 20s may not be needed when you're in your 40s, 50s, 60s. And if you start your illness in your 50s or 60s, you sure are unlikely to need it, the same dose as a 20-year-old. So we've talked about D2 receptors and that they're not altered. What else have people looked at? Well, this is a meta-analysis of all the receptor imaging of the dopamine system that Stefan did with me a couple of years ago, and this is the summary of it. So I've told you there's no alteration in D2 receptor levels. Similarly, if you meta-analyze all the studies of dopamine transporter, there's no alteration there. So I think one or two people thought that might be altered, but it's not. It's a good thought, though. But where you see the major changes in this conversion of dopa to dopamine and the release of dopamine, and actually the levels of dopamine in the synapse, you can measure that, and they are higher in people with schizophrenia as well. What we've also done in this meta-analysis is look at the variability in the measures, and this is summarized here. So the yellow means the variability of the data, a wider confidence interval, if you like, in the data in people with schizophrenia, and that reflects that there is heterogeneity in some of these measures, like the D2 receptor and the DAT, but you don't see that in some of the other measures. So the take-home message is the dopamine abnormality is upstream of the receptor. So this is about the nature of the molecular changes, but what about where in the brain is the dopamine abnormality? And the textbooks all tell us that it's in the mesolimbic system. Is that right? Yep. So here are a couple of textbooks. Stephen Stahl's is there. This is what Stephen Stahl's textbook says. There's dopamine overactivity in the projection from the ventral tegmental area to the limbic striatum, the ventral striatum, nucleus accumbens. And that leads to overactive dopamine and the psychosis. Question, of course, is, does the biology read the textbooks? So I'm going to ask you, are the textbooks right in terms of the mesolimbic system? So your choices are yes, no, don't care, I get all my answers from ChatGPT. So who thinks the textbooks are right? Put your hands up. No one? Oh, one, two? Who thinks no? A few more skeptics. And who gets all their answers from ChatGPT? Marta, and a few others there. Of course, ChatGPT, when it makes up things, they call that hallucinating, which is very interesting, isn't it? Why they set on that term. So are the textbooks right? Well, the other choice I forgot to give you is what's a textbook for the younger people, of course. So we looked at this, and this is Rob's work. He meta-analyzed all the studies. So the more recent PET scanners, you can look at the subdivisions of the striatum, and they're higher resolution. So you could have a look at that mesolimbic part of the striatum, and you can also look at the dorsal striatum, the caudate and putamen as well. And here is the result of the meta-analysis. So anything over 0 means it's higher in people with schizophrenia. And these are the subdivisions of the striatum. The red is the limbic, and you see that actually is not significantly elevated. Maybe there's a hint of something there, but it's not significant compared to healthy people. But the major elevation is in the dorsal part of the striatum, where you get large effect sizes. So an effect size of around 0.7, 0.8 is considered pretty large in medicine. So does biology read the textbooks? Well, it seems not, because actually it's not the limbic system, but actually the mesostriatal, mesocaudate and putamen that is altered. And actually the biology doesn't read the textbooks in other ways as well, because our textbooks all tell us that bipolar disorder and schizophrenia are distinct disorders. But when you look at their dopamine systems, it doesn't seem to be the case. So this is a study that Samir Jauhar did with me, looking at dopamine synthesis capacity in the striatum in people with bipolar disorder, who you see in the black circles, and then also people with schizophrenia with the blue triangles. This is in first episode patients. And it was elevated in both of them, and actually you see that it was related to the severity of their positive symptoms, and the relationship looked pretty much the same in people with bipolar psychosis as it did in schizophreniform psychosis. Quite a strong positive relationship. The highest dopamine synthesis capacity, higher positive symptoms. So this seems to be the case that the dopamine system is linked to the psychosis, irrespective of whether the DSM tells you you've got bipolar disorder or whether you've got schizophrenia. And this wasn't driven by manic symptoms, because we adjusted for it, and you still see that relationship. Actually, this issue of whether the biology reads the textbooks has been tested further, and this is something that Eric Chen, who's going to be talking to you later in the meeting, did with colleagues in Hong Kong, and they're a very nice study that I was also involved in, but they did all the hard work. They looked at dopamine synthesis capacity, again in the striatum, in healthy people in blue, and people with schizophrenia in orange, and you see, as I've told you, yet again, there's that elevation. But then they also looked at people that had a diagnosis of a delusional disorder. You know, this is a psychosis which is just delusions. I don't want to say just delusions because that sounds like it's diminishing it, but they don't have hallucinations and other symptoms. But you still see that elevation in the dopamine synthesis capacity. And then they looked at psychosis NOS, which again is quite a common diagnosis early in the disorder where we're not quite sure what's going on. And again, that elevation was there. So it seems that there is this transdiagnostic role for dopamine. And we've also looked from the prodromal phase to that first psychotic episode, rescanning people, and you see that the dopamine system increases further as people become psychotic. So it seems to change as well. And more recently, we've looked at genetic risk. So these are people that they're not in the prodrome, but they're carrying a genetic marker that means that they're at much higher risk of psychosis than healthy individuals. This is the deletion of some DNA on chromosome 22, which increases your risk of schizophrenia about 40-fold. These are the genes that are deleted. Quite a few genes, including one that we know is involved in dopamine metabolism. But what's very interesting about this deletion is that if you have a duplication of the same region of DNA, it seems that you have a lower risk of schizophrenia. This has just emerged in the last few years. So if you have less of this DNA, your risk is increased. If you have an extra copy of this, your risk of schizophrenia is reduced. So here you have a dose relationship in risk. What about the biology, the dopamine system? Well, we studied this. This is the work of Maria Rogdaki. And here you see, again, dopamine synthesis capacity, and you see healthy people in orange, the carriers of the deletion who have the highest risk of schizophrenia in red, elevated before they've developed the illness relative to healthies, and then carriers of the duplication that reduces your risk in blue. So there's a dose relationship in the dopamine response as well, it seems. So this is all very interesting because it's really all pointing towards a causal role of dopamine in the development and risk of psychosis. But clinically, of course, what do we do? Well, we give people D2, D3 blockers, which is how I think we should talk about antipsychotics. And in my second talk, I'll hopefully give you some reasons why after the break. But a big question then is, of course, well, what do these drugs do to the dopamine system? So I'm going to ask you again, to keep this interactive, what do you think giving someone a drug olanzapine, risperidone. What do you think it does to dopamine synthesis in the striatum to individuals? Does it do nothing? Does it increase it? Does it decrease it? So if you think one, put your hand up now. That's no one. 2, it increases it. Quite a few hands up for that. Number 3, reduce it. Nothing. So again, I should have put another option, which is don't know, don't care. But oh, there are some people on the chat who think it reduces it. Okay. So I think ideally you would think what we want it to do is reduce it, because as I've shown you, this seems to be elevated in at least the majority of people with schizophrenia to some degree, and we'd hope our treatments might correct that dopamine overactivity. Well, this is a study that we did in first episode people. This is what we did, is we scanned them prior to starting an antipsychotic when they were antipsychotic naive. They had the PET scan. You can see that you can measure the dopamine system there. They then went on to their antipsychotic treatment for at least 5 weeks, and then they were rescanned so that we could test whether there was a change. And I have to say, going into this study, I really had no idea. But actually, I would have, if you had asked me, and forced me to bet, I would have said increased it, because of the consequence of blocking the autoreceptor, which is one of the regulatory mechanisms, and we know D2 receptors block that, and that's a feedback loop. So I thought it might maybe have increased it. But here are the data. Absolutely no change. So 20 patients taking an antipsychotic for 3 weeks and no effect. And now I think that's quite a reliable result. We've studied patients that have taken antipsychotics for years, and we also see that the elevation in their dopamine system is still there. So I think it's something that our current treatments don't seem to have much effect on.
Oliver, I understood Alan Young to say this morning that aripiprazole blocks the autoreceptor and increases dopamine release in bipolar disorder. Is that not correct then?
It's certainly, if you block autoreceptors, you do increase dopamine neuron firing. But those studies often are just give the animal a dose, a single dose, have a look at what's going on in the dopamine receptors, or usually it's cell recording, so it's not even an animal. What we don't know very much about is what happens long term. And actually, we have done these sorts of studies in animals, in rats, given them haloperidol as well, and we don't see any effect over days of treatment. Because this is the difference between giving something acutely, a one-off dose, versus, in this case, doing what we do clinically, which is give it repeatedly. And repeatedly, it doesn't seem to have a big effect. Certainly in animals, if you give a single dose of an antipsychotic, the initial response, and this is the work of Tony Grace in the 80s, is that the dopamine neurons start firing a lot. So probably initially there is a response, but over the course of days and weeks, if you take it regularly, the system adapts, or certainly you don't see a change in that we can detect with these measures. So I've been telling you a lot about dopamine, but what does dopamine actually do in the brain, in all our brains? So I'm going to ask you a question. So what answer best captures what mesostriatal dopamine is signaling? So number 1 is rewarding experiences. Number 2, hedonic or pleasure experiences. Number 3, teaching signals. Number 4, alerting signals. So put your hand up if you think number 1. Couple of hands. Number 2. Couple of hands. Number 3. 4 hands, 5 hands. Number 4. 6 or 7 hands, and I should again have given a don't know, don't care option. Put your hand up if you don't know or don't care. There's a few people. Right, well that's very interesting. So alerting was the top choice. Well, let's have a look at the data, and this is a study that Wolfram Schultz published in the late 90s, because I think if you had said rewarding experiences, you would have been right, I think, in the 90s, when that was really very much the focus of understanding of the dopamine system. And it's still very much, I think, what the lay public think of as what the dopamine system does. But actually, starting with Wolfram's work in the late 90s, early 2000s, our understanding has become much more sophisticated. So what he showed in monkeys is that dopamine neurons fire when you are given a reward. So you get that little burst of firing. But actually, they don't reliably do that. If you repeatedly give them the reward and you paired that with a conditioned stimulus, the dopamine neurons don't fire at the reward, they switch to the stimulus. So that's why it's not really about reward. There's another reason why as well. In a minute I'll show you. So now it's actually signaling something is happening that's important, and when you don't get the reward, you get a reduction in the dopamine neuron firing. Now, of course, this is reward, so you would have been right in thinking that dopamine was all about reward in the 90s and early 2000s, but later on people looked at other things, like aversive stimuli, in this case foot shocks, and here you see there's a burst of dopamine neuron firing to a foot shock. So it's not just reward, it's about anything that's going on that's important to the animal. So actually, what we think of dopamine now is much more about signaling something, a teaching signal, that the animal needs to pay attention, something salient is happening. Now that's talking about what dopamine does in terms of learning about the environment, but it also does something else, which is it tells you about the precision of your sensory input. And this was tested in a study that's relatively recently come out in Science, where they tried to make hallucinating mice. And what they did is they had this paradigm where mice had to learn to do something on the cue of a signal. And they used very low intensity sounds. And the reason they did that was it was just at the edge of detection. And mice are very sensitive hearing, but they got the level so that it was just at the edge of it, and they manipulated it to the individual mouse. So then the mice were not sure whether they heard the sound. And what they looked at is the degree to which they then responded. So this is responding. So the mice learnt that if they responded to the sound, they got a reward, so they worked for it. And when it was at a very low intensity, they were getting these false alarms. Sometimes the mouse thought there was a sound when there wasn't. This is the false alarm response, which is what they're looking at. And they call that hallucinations, because the mouse thought it was hearing something, but it wasn't. Now, I'll let you decide whether that's what a hallucination is or not. But it's an interesting model. And they did some computational modeling around this as well. You know, what they were looking at is how long did the mouse spend trying to get the reward when it thought it had heard something when actually there wasn't. And so this was a measure of its confidence in its prediction about the sound having occurred. So I think an interesting study. We can debate whether it's a hallucination. But what they also did, which was very interesting, is that they then manipulated the dopamine system, and they stimulated the dopamine neurons in the striatum. And if you did that, which you can see in purple here, the mice were much more likely to think they'd heard a sound when there wasn't. So these, in quotes, hallucinating mice were made to hallucinate more when their dopamine system was more active. So why am I telling you all this? Well, I'm telling you this because this actually maybe helps us understand what's going on in our patients, because they've got this faulty teaching signal, and perhaps this faulty signal in terms of the confidence of stimuli, sensory inputs as well, when their dopamine system is overactive. So this is overactive dopamine system, more of them firing, and this leads to you thinking that things are going on and thinking that you're hearing things, misperceiving perhaps internal stimuli. So this is my model of how dopamine could lead to psychosis. And this dopamine dysregulation, I think, leads to something that is akin to an experience that a lot of the patients talked to me about when I was working in the prodromal clinic. So these were people that had early subclinical symptoms that we know put them at increased risk of developing schizophrenia in the next 1 to 2 years. And a lot of them would come to me and say, "Doctor, I feel that it's a bit like being in the Truman Show." So for those of you who haven't seen that film, the main character is living on the set of a soap opera, and his life is being broadcast around the world. But he doesn't know that at the beginning of the film. Everyone else on the set is an actor. And he gradually realizes that actually everyone is an actor. The set is just a set. It's not they're not real houses. And a lot of the patients that I saw in this prodromal clinic would say things like, I think that something is going on, I just don't know what it is. I have a strong sense that other people know what's going on, but I can't put my finger on it. And you could see from what I told you about what dopamine is doing in the brain, how that might be what this dopamine dysregulation is leading to. Because your teaching signal has gone awry, and your dopamine system is constantly firing and saying, look out, watch out, something's happening, something important is going on, or there's a sound there, there's something there. So you can see how that might be what's going on in the prodrome, but then of course, at some point, when trying to make sense of it all, it develops into the full-blown psychosis.
So, Oliver, I get really stuck at this point, because if I was terrified, my dopamine would be high. And how do we know that it's not the terror of the illness that's driving the dopamine up?
That's a really good point. So what I did show you was that it's elevated in the prodrome, but it becomes more elevated as people develop the illness. So I think what is going on is that people are starting with a system that is sensitized, more active, because of genetic environmental factors. That leads to altered processing of their stimuli. And there are, of course, other factors that contribute as well. It depends on how you look on the world, as how you interpret that. But if you have certain beliefs about the world and certain prior expectations, you can see how that might lead to paranoid interpretations and psychosis. But of course, what we often forget as doctors and nurses is that psychosis is intensely stressful. If you believe that you are being followed by the CIA or being watched constantly, that must be incredibly stressful. And that, of course, is feeding back on the system. And we know that stress releases dopamine. So you've potentially got a feedback loop here. So I think you're right, actually, that potentially the experience of the emerging psychosis has biological effects that maybe worsen it, if you like, heighten it. Now, I can't tell you that ultimately, if you go far enough back, I can't say that that prior to that prodromal experience, what's going on, except that I can point to those people who were that study of people at genetic high risk, where many of those hadn't any symptoms at all and already showed some dopamine loading, so suggesting that you've got this sensitized dopamine system already. So that's the model, and I've shown you some of the data that supports it. But in the last few minutes before the break, I wanted just to talk about what might lead to this dopamine dysregulation. because this is, I think, downstream of what may be going on. So I've pointed out that the major change in the dopamine system is not in the limbic part, but actually in the dorsal striatum, particularly the part of the striatum called the associative striatum, that's linked to the frontal cortex. Now, why is that interesting? Well, that's interesting because there's lots of data in animal models that shows that this frontal cortex in particular regulates subcortical dopamine. And we tested whether this might be the case in people with schizophrenia in a study where we looked at the volume of the frontal cortex measured on MRI and whether that was related to striatal dopamine, which is on the vertical axis here. And you can see that they're inversely related. So people with lower volumes in their frontal cortex had the highest striatal dopamine. So that suggests, of course it doesn't prove any connection, but it suggests that these two parts of the brain may be altered in schizophrenia in a way that's consistent with that model, that there's something going on in the frontal cortex that's driving the subcortical dopamine system that's suggested by the animal models. But what could cause that? What is going on that might lead to that lower brain volume? Well, this again goes back to an old hypothesis. This was put forward by Erwin Feinberg. This is a photo of Erwin Feinberg in his later years. He actually put it forward in 1983. This hypothesis that schizophrenia is caused by faulty synaptic elimination. So this was another hypothesis that came about quite a long time ago, not in the 70s this time, but the 80s. So I think of this more of a Madonna hypothesis. It's stayed around a long time. I'll let you judge whether it's got new legs. Erwin actually sadly died last year, but he was in time to see some of the new data, and I'm going to share with you some of the data that's just come out from a study that we did. But one of the most exciting things, I think, came out of a lab from Carl Selgren, who's in Karolinska, where he looked at what a cell type called microglia do to synapses. So this in red are microglia from people with schizophrenia, and in black, microglia from healthy controls. And then he looked at the degree to which they were phagocytosing or eating synapses, and you can see that there is increased phagocytosis by the microglia derived from people with schizophrenia. So they're eating up synapses more, and that also translated into lower spine density in the neurons derived from people with schizophrenia. There you can see there's a lot of overlap with healthy people. So that's very interesting cellular work, that there is something going on with the genetics of risk of schizophrenia that carries over into cells that mean that they phagocytose synapses more. Now, how is that? How might you understand that? Well, this is a little clip from Sky News talking about one of the studies. So I'm just going to try and play that, and you can get the Sky News take on it.
Microglia are a major inflammatory cell type in the brain and respond to infection or injury by becoming activated, migrating to the site of injury and removing damaged cells. Experts now believe in schizophrenia sufferers the microglia attacks the good connections between the cells, meaning they are unable to communicate with one another.
So that's the theory, and I think Sky News captured it quite well. But what evidence is there that there is synaptic density alterations in people with schizophrenia? Well, there's lots of evidence post-mortem, and you can see that there's lower spines, these little blobs, in samples from people with schizophrenia, and other markers of synapses are lower in people with schizophrenia on meta-analysis. But until recently, we couldn't actually measure synapses in the living brain. But in the last few years, PET ligands that bind to a protein that's expressed in our synapses have been developed. So this is a protein that's expressed in vesicles that actually are involved in release of neurotransmitters. So that means that we can actually mark the number and availability of these synapses in vivo in patients. So this is the first study that was done. We did this a couple of years ago, looking at this tracer binding in people with schizophrenia. So here in orange you see the levels in healthy people, and in green people with schizophrenia. Do you see lower uptake of this tracer, particularly in the frontal cortex? And here is the brain map showing this pattern. So again, you can see it's particularly in yellow, the reductions in schizophrenia in the frontal cortex. So I was very pleased, actually, that Erwin did get to see this before he sadly died, because this is first direct evidence in living people to support his hypothesis. It's now been replicated by a group in Yale, again in people with schizophrenia. So I think we'll see many more studies trying to understand this in the next few years. But I think this has taken the dopamine hypothesis and linked it up to the cortex. And now the model I'm proposing is a little bit more complicated. And it starts in the cortex with alterations in these immune cells and astrocytes that regulate our synapses, that lead to imbalance in glutamate and GABAergic neurons, potentially to lead to the cognitive symptoms and negative symptoms of the disorder, but also via the projections to the striatum, to the dopamine dysregulation and psychosis, which also feeds back on this system. So this is my updated model that incorporates the dopamine hypothesis as well. And I think it's interesting because it's new targets for treatment. And then after the break, I'm going to talk about some of these that potentially target the glutamate system as well. Just finally, I need to talk about Donald Rumsfeld. So you may be wondering what Donald Rumsfeld has to do with schizophrenia. Well, nothing, apart from the fact that he said there are known unknowns. And I think that's very useful to bear in mind. There are, of course, unknown unknowns as well, but we can't do anything about that. But there are known unknowns. And what we don't know yet is there aberrant synaptic pruning that leads to these dopamine changes? Is this really connected? And something else is whether D2, D3 blockers, they don't work for all people. Antipsychotics don't work for quite a proportion of people. And I think a big unknown is whether there are biological subtypes that are linked to that. So I want to leave you with those thoughts and then just summarize what I've told you about today. So the major dopamine abnormalities, those of you that said dorsal striatum or striatal dopamine synthesis and release, you were right, but it's the dorsal striatum, reliable finding. It's linked to psychotic symptoms. It seems across disorders, although I'm not yet sure about psychosis in major depression. We've got a study ongoing on that. It's linked to lower frontal cortical volumes. So potentially, actually, these biological findings are linked in schizophrenia, and it's not altered by D2 blockers. Remember, D2 receptor levels decline with age, so we should use lower doses of antipsychotics in people over 50. And you may need to increase it if they don't respond, but it really says we should be starting lower than you think. Because, amisulpride at 25 milligrams might be enough if you're in your 70s. And then I've shown you evidence that there's something going on in synapses in people with schizophrenia. And this is really pointing to new upstream targets to treat and even prevent schizophrenia. Why do I think this is all important and interesting? Well, as I've talked about at the beginning, we have a problem in developing new treatments in psychiatry in general, and we need to bridge this translational gap. And I'm hoping that understanding more about the pathophysiology will help us and enable us to make the leap As you can see, I'm not a very good animator, but that's my attempt to show you what I think we need to do. And of course, I've also shown you that the dopamine hypothesis is still alive and evolving, unlike ABBA, who are now just avatars. So in London, you can go and see holograms of ABBA. You don't actually see the real people anymore, but they're just stuck in the 1970s. And in fact, I'm going in July with my daughter, so I'll tell you what it's like. Thank you very much for your attention. I don't know if we have time for any questions.
Well, yeah, we do. Shall we start with someone who hasn't asked any questions? Diego, actually? So this morning wasn't sufficiently interesting to ask this question. I think we usually put the slides on the website. So unless there are particular slides that you
No, I think I'm fine to share the slides.
Yeah. Diego. This is Diego Quattroni, who is a consultant and a senior lecturer at the Institute of Psychiatry, but has also been very involved in putting on the forum.
Thank you, Robin. Thank you, Oliver, for the fascinating talk. I just was wondering this theory of schizophrenia, of the nigrostriatal pathway as the primary dysfunction in schizophrenia, is also consistent with the classic theory of schizophrenia of a primary motor disorder.
That's a very interesting question. So I think we forget, actually, that people with psychosis in their first episode, even before antipsychotic treatment, a proportion of them present with motor signs as well. And there have been nice studies in countries where you're less likely to get antipsychotic treatments, you can do them. So in places like Africa, where you look at the rates of motor symptoms in first episode psychosis, so you know they haven't had an antipsychotic, and it's around 20-25% of patients presenting with motor signs and symptoms as well. Dyskinesias, catatonic symptoms, posturing. What I showed you is that there is alteration in the dopamine system in that putamen part of the striatum as well. So it could well be that is driving the motor problems that our patients present with. I think we tend to have forgotten this because we think of any motor problem as a consequence of the D2 blocker, but there may actually be something there due to the pathophysiology as well.
There's also these wonderful studies of Elaine Walker using videotapes, maybe before your time, Oliver. So she asked parents to send her videotapes of the child who became psychotic and a brother or sister, and the child who became psychotic even at age 2 or 3 had tics and was clumsy and showed motor abnormalities. And the people she showed it to had a high degree of accuracy in pinpointing who was the psychotic. Have I taught you something?
Yes. Well, I didn't know Elaine Walker had done those, but yes, I know there have been studies about these. So yes, again, fitting with this, there's perhaps the dopamine system is vulnerable because of genetic or early environmental factors, and it involves the motor part as well.
Oliver, there is one online, first of all, congratulating for the super talk, and then saying, can you comment on supersensitivity of the receptor in patient in long-standing treatment? Is supersensitivity reversible?
It's a great question. I as I hoped came across, I have to say, that the only data we have is that study that I showed you, the only data I'm aware of anyway. So I don't think we can say that there is definitely supersensitivity caused by antipsychotics. But I think clinically what it means is that if you might need to think in patients as they get older, look out for side effects more, and think about trying to reduce the doses. And if you see that breakthrough psychosis emerging in people, it does say that putting up the dose in the long term is probably a waste of time. So maybe we need to think about alternative strategies to treat them. And clozapine is is the one that we know is effective. And I'm going to talk about that a little bit after the break when I talk about new treatments as well. The other one is the possibility is partial agonists. And we've just finished a study. You'll have to invite me back another year if you want to hear about the outcome. But all I can tell you is we compared amisulpride to aripiprazole in healthy volunteers, and there were differences, but big differences in their effect, in the effects of those two drugs on dopaminergic measures. So I do think that is an interesting alternative approach as well.
And of course, tardive dyskinesia is thought to be a supersensitivity disorder, and if you stop the drug, eventually it can normalize, or clozapine might facilitate it.
Yes, and then treatment for tardive dyskinesia is valbenazine or tetrabenazine. These are dopamine depleters, but working presynaptically. The interesting thing with TD is we've just been doing a study on this, and actually the prevalence of TD, I think, has dropped markedly in the last. We can't find patients with TD, by and large, and I think that probably is because we don't use the same high doses that we used to. So, which is good news, I think.
Hi, my name is Fatma. Thank you for the great presentation. I've actually been involved in a systematic review on this topic in Toronto. My colleagues and I are wondering if you're doing any work in measuring synaptic density in CHR populations or first episode psychosis populations?
We've just finished a study in first episode psychosis, which is under review at the moment, and the interesting thing there is we don't see the same degree of changes. We haven't looked in CHR, or that's the clinical high-risk people, so prior to the onset of the first psychosis. So I think that's also very interesting because it's good news. I'm not saying, I mean, we need to watch this space, but it doesn't look like the synaptic changes are there from birth. At least in our studies so far, it seems that there's something that's different between the first episode and the multi-episode patients.
Hi, I'm Taran Bastin-Pillay from Adelaide, South Australia. Do you have a hypothesis why clozapine might work for supersensitivity psychosis and possibly TD, but counterintuitively also has a rebound psychosis? What would the mechanism from a dopamine hypothesis for that clozapine effectiveness?
So Robin's excited by this because he's been talking a lot about this. We have been looking at the how clozapine works. I've actually been focusing much more on the glutamate system, and we've got some data on treatment resistance and glutamate. I think you could speculate that what we do know is that clozapine doesn't block the D2 receptors to the same degree over time, and probably to the same level even acutely. And if you look at the PET, because it's binds at lower levels and is off the receptor very quickly. So, I think that the speculation is that might allow the receptors a chance to recover.
So we better not ask too many more questions, because Oliver will be using up the second part of his second talk.